Good afternoon. It's an absolute pleasure to be here. I want to thank Aggie and Bill Go and Howard Reber for inviting me not just to be here today, but also to be a member of the Scientific Advisory Board for this very important organization. We have a parallel world over in Michigan where I run the Pancreatic Tumor Program and Multidisciplinary Clinic, and our goal is mutual to try to impact outcomes in patients with pancreatic cancer. So I wear a surgical hat. I'm a card-carrying surgeon, but I also have a basic science laboratory of about 12 people or so where we are tackling multiple aspects of pancreatic cancer. So it's a lab really not focused on a discrete signaling pathway, but really on trying to come at pancreatic cancer from multiple angles. Today I'm going to talk to you, is that a crackling fire back there? Never thought I'd see that at UCLA. I'll have to tell all my friends back home. Today I'm going to talk to you about some work that our laboratory has embarked upon really in the last maybe about 4 years or so, trying to address a new hypothesis in the field, and that being on cancer stem cells. And for those of you who missed the morning session, we had a lot of slides about, the numbers with pancreatic cancer. We're all very familiar with those, and trying to come about the disease at different angles. And so this is one program within my laboratory that's trying to come at pancreatic cancer from a different angle, and that being addressing the cancer stem cell hypothesis. So what is this hypothesis? It's really that not all cancer cells are the same, but that cancers are driven by a subset of cells within a cancer that are primitive and have stem cell-like features and are the drivers for the cancer. I think we have good data to support this hypothesis. I'll show you where we started and where we are now and where we hope to go. This is a cartoon that was actually published a number of years ago that suggested this hypothesis might be the case. And on the left is the general thinking about how cancer evolves, where that all cancer cells, even though cancer cells may be diverse, that all cancer cells are capable of forming cancers, while the cancer stem cell hypothesis on the right suggests that it's really a unique subset of cells within cancer. Not to make things too complicated, but it's probably a hybrid of both theories, but certainly we think that the cancer stem cells have a unique feature within cancers to be the drivers. When we started working on this area, it was really based on a key paper that was published in, it's actually published in Michigan, looking at this concept in solid cancers for the first time, and it was actually in breast cancer. And I remember seeing that paper, and a week later, wanting to embark on exploring the concept in pancreatic cancer. But really, to look at it in pancreatic cancer, you had to have the right model systems, and it was really patient tumors. And so we set about developing what we call a xenograft program in the laboratory, where we take patient tumors, and these are usually patients who undergo surgical resection, and we either study them directly, and that's where it says primary, but most of the time we don't have enough material directly from the pathologist, and so we grow them in immunodeficient mice and expand the amount of material we have so that we can try to do critical studies. And this program or platform actually is, I think, a very important one to have to look at human pancreatic cancer biology. It took about 9 months or so to get the program up and going, and I'll talk a little bit more about where the program is now, but we use this basic platform to digest pancreatic cancers, and probably most of you haven't felt a pancreatic cancer in your hand, but they're hard as a rock, and so we worked on optimizing the techniques to digest the tumor so we could have viable cancer cells at the end of it, and then we sorted the cells based on different cell surface markers that we thought might be important in identifying stem cells, and then implanted the cells and looked at their ability to grow tumors and also at their biology. And this was our first paper exploring the concept of identifying cancer stem cells in pancreatic cancers, and the markers, which you can see up here on the left, were the markers we initially chose based on, again, work that was done in breast cancer. And you can see that each of these markers is expressed in only a subset of pancreatic cancers, and then just by doing various combinations and looking at tumor genicity, we came to find that this bottom population here, highlighted in red, was a very highly tumorigenic population and generated tumors and had stem cell-like function. And this was based on analysis of 10 different tumors. And this is just a typical experiment looking at this where you, here, these are all cancer cells. If you take cells that express this cancer stem cell-like phenotype or marker profile shown on the right, that those readily made tumors, and that cancer cells that didn't express those markers did not. And then this is the patient's tumor, and here you can see the tumor. It's a little bit hard to move this mouse around here, but you can see the tumor that was generated from these cancer stem cells look just like the patient's tumor. So these cells are able to regenerate themselves and also to generate all the phenotypic heterogeneity that we see in a patient tumor. We did a lot of that work. That one paper I showed you was about 2.5 years worth of work of really looking at differential tumor growth and the biology. And those were tumors cells that we implanted in under the skin of the mouse, but we thought it was important to validate the concept by actually putting the tumor cells in the pancreas. And this is just an experiment showing that, where we take our cancer stem cells, if you will, shown here, and found that they readily formed tumors in the pancreas, while the pancreatic cancer cells that did not express these markers did not, kind of validating our concept in the appropriate niche in which pancreatic cancer develops, that being the gland of origin. This is kind of a complicated slide for those who aren't used to looking at FACS plots, but really the main point I want to make is that it's not just about tumorigenicity, but stem cell function. So can cells self-renew or make themselves? And just to highlight that, if you just look at the bottom, if this is the patient's primary tumor, and this population in the box is the cancer stem cells, which we isolate here, you can see that when we implant these, that they not only are able to generate themselves, but all the progeny that are marker negative, just like we see in the primary tumor. And that's an important concept in defining cancer stem cells. When we did that initial work, there's been subsequent work using different markers to identify cancer stem cells. And while our population that we had was highly enriched, we didn't think it was a pure population. And so we set about trying to see if there were other cell surface markers that might be helpful in further refining our cancer stem cell population. I list 3 here. ALDH, which is proved to be a good marker for cancer stem cells in the breast. CD133, important in identifying cancer stem cells in the colon and brain. And then in the middle, CMET, and we explored this because there were a couple papers that came out around the time we were doing this that showed that actually CMET might be a stem or progenitor cell marker in the normal pancreas. And you can see that again, these markers are just expressed in a subset of pancreatic cancer cells, again highlighting that not all cancer cells are the same, and that cancers are very heterogeneous. And these are some tables that get at the issue of how many of these cancer cells do you have to implant to get a tumor. And here we go from 100 cells up to 10,000 cells, and then we sort based on the different markers. And what we found using these different markers is that actually CMET alone was pretty effective in generating tumors, and we didn't see tumors form until we injected at least 10,000 cells that were CMET negative. And that appeared to be very favorable compared to what our original cancer stem cell population was. And interestingly, you can see some of these other markers, like for example, look at ALDH here. It has less of ability to make tumors, and then in the negative population, we're starting to get tumors at much lower numbers, suggests it's not as effective in its tumor initiating capacity. And then we went on to look at a combination of CMET and CD44. Data in our lab suggests that these 2 markers might be the most powerful, and in fact, it proved to be the case, where we began to see tumors with as few as 50 cells. So right here, this data suggests that this is a better marker profile, if you will, for pancreatic cancer stem cells. And I also want to highlight that this profile we're defining here is different than is seen in breast cancer and brain cancer. So while there are some overall common concepts in cancer stem cell biology across tumors, there are some differences among tumor types. In addition to identifying markers to pick out cancer stem cells, we wanted to see if we might be able to use this profile to actually target pancreatic cancer stem cells, and we turned to this simpler assay. It's called an in vitro tumor sphere assay. So it's been shown that when people want to try to find stem or progenitor cells in normal tissues, for example, this assay was developed when people were trying to find stem cells in the brain, that if you take cells and you have them float in media in a dish, only cells that have self-renewal capacity can make a ball of cells, so it's called a sphere assay. And what we found is that the cells that had a cancer stem cell-like profile would readily make these balls of cells or spheres, while the non-cancer stem cells would not. And interestingly, what we have found is that this is a pretty good quick and easy screen, this in vitro screen, as opposed to the previous data I showed implanting human tumor cells in immunodeficient mice. That's very time-consuming and costly. While that remains the gold standard, we're interested in finding ways to do more high-throughput work, and this sphere assay has proven helpful. And then this is just some basic work looking at whether targeting c-Met might be useful in affecting pancreatic cancer stem cells. And what you can see here in the top left is cells that express c-Met readily make these spheres, while cells, again, these are all cancer cells that don't express c-Met are much less effective. Here, if we take a drug that actually is in clinical trials, a c-Met inhibitor, it's being used currently in phase 1 clinical trials, that it's effective in inhibiting sphere formation in a dose-dependent way, and the doses that work are the doses that knock down the c-Met pathway, as shown over here. And what we found is it looks like c-Met inhibition causes apoptosis in the cancer stem cell population, as shown here, preferentially above the bulk cancer cells. And then importantly, we wanted to test whether it was effective in vivo. And so this is an experiment. We took 3 different patient tumors, and we grew the tumors, and then we treated with different agents, either untreated or treated with the c-Met inhibitor, this XL184, gemcitabine, or the both. Here we treated for 4 weeks and then stopped treatment. And what we found is that all of these treatments were effective in blunting tumor growth. And the combination of the c-Met inhibitor and gemcitabine was most effective. But interestingly, if you look at these tumors that were harvested and looked at the cancer stem cell population, what we find is even though gemcitabine can have some effect in inhibiting tumor growth, it really didn't touch the cancer stem cell population, while the c-Met inhibitor was effective in knocking that down. And I think and I'll show you more data, but I think our overall belief is that we're going to need to have a drug in the armamentarium of what we give patients that's going to be able to target this cancer stem cell population to really provide a cure for patients. Now, this was an interesting experiment we did based on an overall concept that cancer stem cells may be important for metastasis, and to try to get at that question independent of effects of the inhibitor affecting primary tumor growth, we set up a metastasis model, and there are several different types to choose from. But in this case, what we did was actually inject cancer cells into the heart, and then 24 hours after, we either left the mice untreated, treated with standard chemotherapy, or with the c-Met inhibitor. And we treated for 2 weeks, and then we analyzed the animals 6 weeks after the injection. And what we found is that there was a very modest effect with standard chemotherapy, but actually the c-Met inhibitor was very effective in completely preventing the development of metastasis. This was not just seen with luciferase, as you see here, but we actually sacrificed the animals and in a painstaking way went through lung, liver, and other organs to look for any viable cancer cells, and were not able to find any. And so what I think this tells us, that if we come up with therapies to target cancer stem cells, they may be in particular very effective in the setting of either the neoadjuvant setting before we resect patients, or even the adjuvant setting, because we know when we do a surgical resection, there's a 75% chance that the patient's tumor is going to recur. And so we think that some of these strategies might be in particular useful in that setting. And then I just thought I'd touch on this issue. So I've shown you a lot of this data about different markers, and I think to do these kind of assays on biopsies of pancreatic cancer is not easy. So can we move beyond looking at cell surface markers and actually try to get a cancer stem cell signature, if you will, so we can really understand what the critical pathways are? And this is a little bit of a complicated slide, but I just want to explain the concept. This was actually based on work done by John Dick at Toronto, where he did a similar type of experiment in leukemia. So the markers may have some variability depending on the individual tumor, but what he did is he did limiting dilution assays where he validated the tumor, the cells in each patient's leukemia that were cancer stem cell-like, and then did gene expression profiling of 16 different leukemias, and was able to find a gene signature that interestingly overlaps significantly with stem cells in the blood, hematopoietic stem cells. And so, and importantly, what they found is that gene signature really correlated with patient survival. So this was important data because it shows us it's not just a marker, but it actually, or that cancer stem cells aren't just arbitrary, but they actually do relate to patient survival. And so we've tried to take our work to the next step to try to get a sense of how do cancer stem cells affect patient survival. So this is, we're just starting on this, but since we've had that xenograft program up and going now for a number of years, we actually now have 30 different patient tumors that we're analyzing, and we find there is significant heterogeneity amongst tumors. And this is just an analysis of 20, but it still is a lot of work. What we've done is take 20 different tumors, and we looked at all the cancer stem cell markers that I've shown you across those 20 different tumors. And we want to have some understanding of the relationship of the expression of these markers and the tumor's biologic activity. And to do that, we're looking at tumor growth rate and ability to metastasize. And this is a busy slide, but I just want to distill it down for you. So if you just look at the top, here are all these different markers I talked about. And then here are these 20 patient tumors. And this was just a couple weeks ago. We laid all this data on this big table, and I said to everybody in the lab, I said, you know, what do you guys think? You know, what do you see? What are the trends? And there was one thing that really stuck out. We have 20 different tumors, and they all act a little bit differently, but we have 3 tumors that are incredibly aggressive. And these 3 tumors had very high percentages of CD44 c-Met, and they're highlighted here in red. This J tumor, this one right here, and this one. And the other markers really did not seem to correlate with biologic aggressiveness. Now, we haven't done metastasis experiments on all of these, but we do have a good sense of the growth rates. And so this is really our first sense of how these profiles might correspond with biologic activity. And this is just a couple of FACS plots from some different tumors. So most of the tumors are actually in one of these categories where the CD44 c-Met expression is either very low or a little bit higher. But here are 2 of those really 3 bad-acting tumors, and I can tell you, because I resected these patients' tumors, these patients had a very quick return of disease. And so these 3 tumors had very high levels of CD44 c-Met. And so we categorized the different patient tumors based on low, medium, and high expressors, and that's just shown here on this table. And the ones that are high expressors are very aggressive, and the ones that are low expressors are much slower growing. So what our next step is to actually do gene profiling of the CD44 c-Met populations in the high and low growing tumors to try to get that gene profile and try to get a better handle on globally how to go after these cancer stem cells. And so these are the experiments underway. We're doing limiting dilution assays of the CD44 c-Met cells, try to get a gene expression profile of the cancer stem cell signature. And another important piece of this is if we have these 20 tumors and we know their biologic activity, we're going to also be doing sequencing of those tumors to look at the genetic mutations and try to get some understanding of the mutational profile in that tumor and the cancer stem cell profile and its biologic aggressiveness, because ultimately we want to be able to just take a biopsy of someone's tumor and have an understanding of how to target it and how the relationship of the mutations corresponds with the cancer stem cell function. I thought that I would just mention some of the work we've done looking at these two other signaling pathways. These are two developmental signaling pathways that are thought to be important in normal stem cell function and are being explored as targets for cancer stem cells. And importantly, I thought for this audience, they're actually drugs targeting these different pathways that are rolling out in the clinic. And so I thought I'd share with you where we are in exploring inhibiting Notch and Hedgehog signaling. So first, just a little bit about Notch. Notch is actually important in the pancreas. It is required for expansion of undifferentiated pancreatic progenitor cells during development. Normally, Notch signaling isn't active in the adult pancreas, although it is increased during pancreatic cancer development and progression, and so we thought it might be a valid target to go after to hit pancreatic cancer stem cells. And again, this type of slide looks familiar, and this is our initial screen, is targeting Notch signaling. And here we use a drug called gamma secretase inhibitor, and it targets a key part of the Notch signaling pathway. And here you can see the results look favorable, that using this gamma secretase inhibitor is able to knock down sphere formation. And counter to that, if we actually use a Notch ligand, that actually promotes sphere formation, supporting the concept that Notch signaling is important in the function of these cells. And so we, again, went on and did our in vivo experiments, again, using gamma secretase inhibitor, gemcitabine, or both. And we saw that all the treatments had some effect on tumor growth, as shown here. And that gamma secretase inhibitor was effective in knocking down the Notch signaling pathway as measured by HES1, which is a critical transcription factor target. But that gamma secretase inhibitor was also effective in knocking down the cancer stem cell population. And again, gemcitabine is not. And so where we are with this is we've actually moved this work into clinical trial. This is a clinical trial for patients who have resectable pancreatic cancer. It's a single-arm, multi-institution phase 2 clinical trial where we're planning to treat 30 patients, where they're going to get the Notch inhibitor for 2 weeks, and then they're going to have a resection on week 3. So here we're going to have the tumor in our hands so we can look at the effect of targeting Notch signaling and the effects on the cancer stem cell compartment, and we've powered it so that when we use control patients who haven't been treated, we'll be able to get a sense of the effect of the drug on cancer stem cells, and importantly also on hitting the pathway. So that's just one example of taking that very basic work we've done and moving into the clinic. And of course, that's an important part for us because I take care of patients, and patients are constantly wanting, there's lots of neat things we can do in the laboratory, but we have to keeping our eye on the ball, which is trying to move things forward to the clinic and trying to improve outcomes for patients. Here's some data on sonic hedgehog. Again, it's a pathway that's been in the news a lot lately. This was from our original paper back in 2007, where we showed that the cancer stem cell population, that's on the far right there, actually produced a lot of this ligand. And actually what's been found is that sonic hedgehog is really important in promoting stroma activation in pancreatic cancer. And so this led us to believe that perhaps pancreatic cancer stem cells are important in promoting pancreatic cancer by enhancing the stroma. There is a Hedgehog antagonist. It's actually available in clinical trial, and that we tested it, again, the same kind of experimental paradigm. And interestingly here, what we see is that the Hedgehog inhibitor alone doesn't have any effect on primary tumor growth, although when you combine it with gemcitabine, it has an additive effect. But here's an important finding, and again, that's why you have to look at different aspects of what therapeutics might do. We've tested the drug in a couple of the tumors that are highly metastatic, and while it didn't affect primary tumor growth, it actually has a pretty dramatic effect on preventing development of metastasis. So the arrows there on the top and on the bottom show the primary tumor, and they're really unchanged in size, but you can see treatment has a pretty dramatic effect on inhibiting metastasis. And here was an interesting thing. You know, there was a paper that was published in Science probably about 2 years ago now that showed that targeting Hedgehog was really important in ablating the stroma, decreasing the stroma. And we do see that in these mice, but we also see something else interesting in that the Hedgehog antagonist also seems to change the appearance of the cancer cells. They seem to possess more mucin-like material, and actually when we do RT-PCR for a number of genes, it looks like it may induce some form of differentiation. And so that's something that we're currently exploring in the lab. And even, we don't think the Hedgehog antagonist is working directly on the cancer stem cells. We have some data that shows us that, and it's also been shown by some other investigators recently, but we think that it's affecting the niche in which the cancer cells exist. And here is an experiment in which we measured the cancer stem cells after treatment. You can see the cancer stem cells are actually decreased in numbers after treatment with the Hedgehog antagonist. And we think that it's affecting the niche in which they're existing and causing the cancer cells to undergo some form of differentiation, but that's something we're currently exploring. And so just where this in vivo preclinical work stands, we know that the Hedgehog antagonist affects the target genes in the mouse stroma, but not in the human cancer cells directly. We're exploring whether the Hedgehog antagonist might affect the cancer stem cell function, and by that we're reimplanting the treated cancer cells. But this interesting work has led us to move forward to a clinical trial to test the effects of Hedgehog antagonists in patients with metastatic pancreatic cancer. And this is the clinical trial, and ideally I showed you data that showed the drug looked like it might be very effective in preventing the development of metastasis, but the only way we could actually get the drug was to treat patients with advanced disease. And I won't go into that right now, but just so, and I'm sure many of you are aware, the clinical trial process is a little bit skewed in this country. Sometimes you really can't use the drug in the lab to test how to best use it until the clinical trials are already happening. Nevertheless, in this case, we have this clinical trial, and believe it or not, it's a first in its kind where we're actually treating patients with metastatic disease. It's a single institution pilot study, 25 patients where we're doing 3 core biopsies of either the pancreatic tumor or metastatic lesions before and after treatment with a Hedgehog antagonist. And then gemcitabine is being added on following. And the primary goal of the clinical trial was to try to understand how Hedgehog inhibition might affect cancer stem cells and Hedgehog signaling. And obviously, our overarching goal, although they're called secondary endpoints here, try to improve clinical outcomes. And believe it or not, this is the first clinical trial. I'm almost done. And where we have paired biopsies before and after treatment, what I think highlights part of the problem with understanding what we're doing in the clinic and treating the disease. And here's an example of one of the patient biopsies where we have pre and post treatment. And interestingly, you can see that we're seeing some of those changes in the cancer cells, just like we saw in the preclinical samples. And so this is really the reason why we got into this business in the first place. It's based on this cartoon that's been around for the last 5 or 6 years, suggesting that cancer stem cells are resistant to standard therapies. And right now we're giving our patients standard therapies, but we need to move forward beyond that and try to figure out how to target this population more effectively. I'm just going to slide forward a little bit. So my last slide. Where are we now? We're testing promising therapeutics. I mentioned some of the pathways we're going after. We're also trying to go after it in an unbiased fashion by doing gene profiling of cancer stem cells to figure out what pathways overall might be best to target. And importantly, we're also interested in trying to understand the contribution of the tumor microenvironment on pancreatic cancer stem cell function. Is this a fixed population, or are there signals between the tumor microenvironment and the cancer stem cells that may affect their function? Important, I want to highlight here, we have multiple different model systems up and going in the lab, not only human xenografts, but also genetically engineered mouse models, 3D culture systems in place. And with that, I'll wrap it up, and importantly want to highlight all the key people in my lab and collaborators that have helped with this work. Thank you.